Hello students. Today we are starting a new chapter, Strategies for Enhancement in Food Production, which comes under the unit Biology in Human Welfare. With ever increasing population of the world, enhancement of food production is a major necessity. Biological principles as applied to animal husbandry and plant breeding have a major role in our efforts to increase food production. Several new techniques like embryo transfer technology and tissue culture techniques are going to play a vital role in further enhancing food production. Let's have a look into animal husbandry. Animal husbandry is a branch of agriculture which deals with systematic practice of breeding and raising livestock by applying scientific principles. It includes the care and breeding of buffaloes, cows, cattle, sheep, goats, camels, pigs, bees, and silkworms. It also includes fisheries and poultry. Humans rear animals for various products like milk, egg, meat, silk, wool, honey, etc. It is estimated that more than 70% of world livestock population is in India and China. However, it is surprising to note that the contribution to the world farm produce is only 25%. That is, the productivity per unit is very low. Therefore, in addition to conventional practices of animal breeding and care, new technologies also have to be applied to achieve improvement in quality and productivity. One of the most important aspects of animal husbandry is animal breeding. Animals with very good genetic traits are mated together in order to improve the quality of future generation. The chief objectives of animal breeding are increased quality and quantity of yield, higher growth rate of farm animals, better assimilation efficiency of reared animals, strong resistance to various diseases, longer productivity, higher acceptable reproductive rate. Let's have a look into the types of animal breeding. They are inbreeding and outbreeding. Watch this video to understand more about animal breeding.
Animal breeding is broadly classified into two types, inbreeding and outbreeding. Inbreeding. We have learnt that the first generation of crossbreed hybrid will have recessive allele in their genome. So in order to have pure hybrid with no recessive alleles in it, inbreeding is performed. In inbreeding, very closely related animals, generally brothers, cousins, etc., are mated with each other. In each generation, the animals that express recessive or harmful character are eliminated, and the remaining are bred again. After repeated inbreeding and selective selection, pure lines of hybrids with no recessive characters are obtained. However, there is problem with inbreeding. When the pure breeds are repeatedly inbred, after few generations, some of the dominant characters lose their effectiveness. This is also called inbreeding depression. To overcome this inbreeding depression, outcrossing is performed. Outbreeding. Outbreeding is a technique in which breeding is done between unrelated animals. Here, the term unrelated refers to the animals of either same breed having different ancestors or animals of different breeds or different species. Outbreeding is generally carried out by three ways. These are outcrossing, crossbreeding, and interspecific hybridization. Outcrossing. In this method of breeding, mating of animals is allowed within the same breed. Here the animals are selected in such a way that both the animals do not have common ancestry for at least 6 generations. By doing this, inbreeding depression can be overcome. Outcrossing infuses vigor and vitality in the offspring. Due to this, outcrossing of pure animals is preferred over impure animals. Crossbreeding. In crossbreeding, two animals of different breeds but of same species are mated together. The offspring produced will have dominant characters of both animals. Crossbreeding is done to develop new superior breeds and also to improve the quality of local breeds. The hybrids thus obtained have more commercial value. For example, when indigenous cow breed Tharparkar having properties like high disease resistance and low milk yield is mated with an exotic breed Holstein having properties like low disease resistance and very high milk yield. A special hybrid was formed called Karen Fries. This cow was both disease resistant and had high milk yielding capacity. In the first generation, there will still be some recessive characters. So, the hybrid is subjected to inbreeding and selection to develop a stable hybrid. Here are some examples of cattle crossbreeds. Several factors are considered before selecting cattle for crossbreeding. Some of these are as follows: quality of breed and its yield value, diet requirement of the breed, ratio of yield to diet, reproductive maturity of the breed, duration of reproductive period, lactation, mammary system, size of udder, nature of the breed, disease resistance, acclimatization, vigor and vitality, capacity to work. Crossbreeding is also done to sheep. Haisadale sheep developed in Punjab by crossing Bikaneri eves and Merino rams. Crossbreeding is also done to improve the poultry breeds. Here the best egg layers and broilers are allowed to crossbreed by a method called pen mating. In this method, one male is kept with 10 to 15 females in rooms called pens and allowed to mate. Here are some of the examples of poultry crossbreeds. Interspecific hybridization. Interspecific hybridization involves mating of male and female of two different species. The offspring has desires characters of both the species. For example, when male donkey is mated with female horse, the offspring is called mule. Mule can survive in harsh environments and are capable of doing hard labor. Due to this, they are considered as good beasts of burden. Here are few examples of interspecific hybridization. There is one big problem with interspecific hybridization. That is, the hybrids are sterile. However, due to their high economic benefits, they were bred regularly.
Let's see the importance of controlled animal breeding techniques. Animals mate when they are in heat. When females are in heat, the availability of superior quality male in that place is rare. Sometimes, even if the female is in heat, the male rejects the female and mating does not occur. Further, the male animals have limited potential to mate. The cattle bulls can mate only 50 to 100 times a year. Moreover, the exotic breeds require a suitable climate for mating that is not present everywhere and every time. Natural mating sometimes accompanies contagious diseases as the males are generally left in open to roam about and are not fed properly. To overcome these problems, controlled breeding using artificial techniques are carried out. Let's watch this video to know about controlled breeding techniques.
There are two artificial breeding techniques: artificial insemination, that is AI, and multiple ovulation embryo transfer technology, that is MOET. Artificial insemination. In artificial insemination, semen from superior quality males is collected by artificial techniques. The semen collected may be used immediately or can be stored in frozen form in cryogenic semen banks. When females are in heat, required amount of semen is injected directly into their reproductive tract. Natural fertilization takes place and the female becomes pregnant. Here, freezing semen is a very good technique as it is easy to transport the semen in a frozen form to the place where the female is housed. There are certain benefits of artificial insemination. These are: semen can be preserved and sent to all the insemination centers throughout the country. By this method, it is possible to mate different breeds of animals, even if certain breed is not present in that geographic area. By adapting this method, farmers can avoid rearing exotic males in their farms, as semen procured from a single male is sufficient enough to inseminate a few hundred females and is economical. This technique reduces the chances of encountering contagious diseases also. However, the success rate of crossing mature male and female animals is low, even though artificial insemination is carried out. Multiple ovulation embryo transfer technology, MOET. In this method, a superior breed of female is administered follicle-stimulating hormone, that is, FSH. This induces follicular maturation and superovulation, where 6 to 8 eggs are produced per cycle instead of one. This female is known as genetic mother. The female is then allowed to naturally mate with superior quality male, or it is artificially inseminated. All the eggs fertilize, and when the fertilized embryos are 8 to 32 cell stages, they are recovered non-surgically and transferred to surrogate mothers. The genetic mother can again be induced to superovulate and produce new embryos for surrogation. This technology has been demonstrated for cows, sheep, rabbits, buffaloes, mares, etc., as more than one zygote can be implanted into the mother. This technique is very useful to increase the herd size in short time.
In this session, you have learned about the importance of animal husbandry and various techniques of animal breeding. With this, we come to the end of today's session. Thank you and have a nice time.